day! Welcome to Teacher Sally YouTube channel. In today's video lesson, I will discuss about other type of non-Mendelian pattern of inheritance, and that would be multiple allele and polygenic traits. I'm here to help you solve genetic problems related to multiple alleles, especially in blood types. Are you ready? So without further ado, let's get started. Learning objective: infer the unknown phenotypes of individuals on the basis of the known phenotypes of their family members and solve genetic problems related to multiple alleles. Let's recall. In previous lesson, you have learned that in codominance, the phenotype of the heterozygote exhibits both the phenotype of the homozygous parent. Thus, the trait is neither dominant nor recessive. In this new lesson, you are going to learn that there are times when there may be more than two types of alleles that control a certain trait, and this certain situation leads to the expression of more than two phenotypes. Let's explore. Sometimes there are more than two alleles present in the gene pool for a gene. For example, blood type consists of two dominant and one recessive allele in humans. Coat color in rabbits is determined by a single gene that has at least four different alleles. Different combinations of alleles result in the four colors you see here. In humans and other organisms, there are traits that have three or more different types of alleles or genes. Whenever a given trait has three or more different alleles, we say that the trait has multiple alleles. Let's elaborate. What is multiple allele? Multiple allele is a type of non-Mendelian inheritance that involves more than just the typical two alleles that usually code for certain characteristics in a species. With multiple alleles, that means there are more than two phenotypes available depending on the dominant or recessive alleles that are available in the trait, and the dominance pattern the individual alleles follow when combined together. Alleles are alternative form of a gene that controls a certain trait. Normally, a gene is controlled by only two alleles, but there are genes that have two or more alleles. Even though three or more alleles exist for a particular trait, an individual can only have two alleles, one from mother and one from the father. Let's have an example of multiple alleles. The color of the skin in rabbits is influenced by a series of multiple alleles. There are four types of coat color in rabbits, and each type denotes specific alleles. The normal color of the skin is brown. Beside it, there are white races called albino, Himalayan as the mutant races. The Himalayan is similar to albino but has darker nose, ear, feet, and tail. The mutant genes albino and Himalayan occupy the same locus and are allelic. Both albino and Himalayan are recessive to their normal allele. In the table, it summarizes the four colors in rabbit. First column indicates the phenotype, second column indicates the allele, third column the possible genotypes, and the fourth column order of dominance. Full color or brown rabbit is represented by capital letter C, and here's the possible genotype. And full color brown rabbit is dominant over all others. Chinchilla is represented by lowercase c with ch superscript, and here's the possible genotype, and it is dominant over Himalayan and albino. Himalayan is represented by lowercase c with h superscript, and here's the possible genotype. So this is dominant over albino, and the albino is represented by lowercase c, so here's the possible genotype, and it's recessive to all others. Number 2 is blood type. The human ABO blood type is a good example of multiple alleles. Take note that there may be multiple alleles within the population. but an individual can have only 2 of those alleles. Why? Because an individual can have only 2 biological parents. We inherit half of our genes or alleles from mother and the other half from father. So we end up with 2 alleles for every trait in our phenotype. Blood type exists as 4 possible phenotypes or blood groups: A, B, AB, and O. There are 3 alleles for the gene that determines the blood type: IA, IB, and small I. You can have just 2 of the 3 blood types in your genotype, 1 from the mother and 1 from father. Number 3 is hair color. Too many alleles exist to count. There are over 20 different shades of hair color. Now let's have the ABO blood system. It is determined by the type of antigen present on the surface of red blood cells. Antigens are substances that are recognized by antibodies produced by the immune system. The I designation here stands for isoagglutinogen, another term for antigens. Those are names for alleles involved in the ABO blood group system. The I stands for isoagglutinogen, which is an antigen that causes agglutination of blood cells when reacting with a specific antibody, an isoagglutinin. The first column here shows the genotypes and the second column shows the phenotypes. Heterozygous IBI for blood type B and 2 small i's for blood type O. Take note, blood type A and B have 2 possible genotypes, homozygous and heterozygous. And blood type AB and O only have 1 genotype each. Now the genotype can also be represented in this way. Based on the table, alleles A and B are dominant over O. Blood type O can be expressed as homozygous, OO, and blood type A and B can be homozygous or heterozygous. In blood type AB, both alleles are codominant with each other. We have here blood type A. It can be represented by homozygous dominant AA and heterozygous dominant AO. For blood type B, it can be represented by homozygous dominant BB and heterozygous dominant BO. And for blood type AB, it can be represented by AB, and for blood type O, it can be represented by OO. Let's have the rules for blood type. Remember, A and B are codominant. The genotypes can be written like this: AA or IA, the one with superscript. And this is homozygous dominant for type A. BB or IB is homozygous dominant for type B. AB or IB is homozygous dominant for type AB. And A and B are dominant over O. AO or IAI is heterozygous dominant for type A. BO or IBI is heterozygous dominant for type B. And OO or II is homozygous recessive for type O. Now, do you know what blood type is safe for you if you need a transfusion? Humans can have red blood cells that are of type A, IA, type B, IB, or type O, I. These 3 different alleles can be combined in different ways, following Mendel's laws of inheritance. The resulting genotypes make either type A, type B, type AB, or type O blood. Type A blood is a combination of either 2 A alleles, or 1 A allele and 1 O allele, IAI. Similarly, type B blood is coded for by either 2 B alleles, IB, IB, or 1 B allele and 1 O allele, IBI. Type O blood can only be obtained with 2 recessive O alleles, II. And type AB blood is an example of codominance. The A allele and the B allele are equal in their dominance and will be expressed equally if they are paired together into the genotype IA-IB. Neither the allele A or the allele B is dominant over each other, so each type is expressed equally in the phenotype, giving the human an AB blood type. Blood containing red cells with type A antigen on their surface has in its serum or fluid antibodies against type B red cells. If in transfusion, type B blood is injected into persons with type A blood, the red cells in the injected blood will be destroyed by the antibodies in the recipient blood. In the same way, type A red cells will be destroyed by anti-A antibodies in the type B blood. Type O blood can be injected into the persons with type A, B, or O blood unless there is incompatibility with respect to some other group system also present. Persons with type AB blood can receive type A, B, or O blood. Take note that the table shows type O blood may be transfused into all the other types and known to be the universal donor, and the type AB blood can receive blood from all the other blood types and known to be the universal recipient. Now, how is a person's blood type determined? Blood types are determined by the presence or absence of certain antigens or substances that can trigger an immune response if they are foreign to the body. Since some antigens can trigger a patient's immune system to attack the transfused blood, safe blood transfusions depend on careful blood typing and cross-matching. When an incompatible blood enters a person's body, the immune system produces antibodies to fight the blood cell. Antibodies bind to the A, B, and or Rh proteins on the surface and cause the blood to clump. This clumping is called agglutination. Blood types are determined by mixing a blood sample with antibodies to see if clumping occurs. Let's have sample problem. Number 1. Identify the possible blood types of the offspring if a woman whose blood type is O and married to a man whose blood type is AB. Step 1. Identify the genotype of the parents. AB cross 2 OO. Step 2. Construct the Punnett square. Put the gametes of one parent on top and the gametes of another parent along the side, and show the possible outcome of the cross. Step 3. Interpret the result. So we have 2AO or blood type A and 2BO or blood type B. Step 4. Write the genotypic and the phenotypic ratio of the offspring. So we have genotypic ratio of 2:2 or 50% heterozygous dominant AO and 50% heterozygous dominant PO. And the phenotypic ratio of 2:2 or 50% blood type A and 50% blood type B. The possible blood type of their children are type A and type B. Number 2, show the cross between a mother who has type O blood and a father who has type AB blood. Put the gametes of one parent on top and the gametes of another parent along the side. Then show the possible outcomes of the cross. Interpret the results and write the genotypic and the phenotypic ratio of the offspring. So for the genotypic ratio, we have 50% IAI and 50% IBI. So the genotypic ratio is 2:2. And the phenotypic ratio is 2:2 or 50% type A and 50% type B. This is heterozygous type A, heterozygous type A, heterozygous type B, and heterozygous type B. Number 3, if a man with AB blood has children with a woman who has type O, what will be the possible genotypes of their children? What will be their blood types? So you have to identify again the genotype of the parents and construct the Punnett square. We put the gametes of one parent on top and the gametes of another parent alongside and show the possible outcomes of the cross. we have your IA, IB, and two small i's. Then IAI, heterozygous type A, heterozygous type B, heterozygous type A, heterozygous type B. Number 4, a woman with type B blood has a child with type O blood. How is this possible if her husband has type A blood? So we're going to use heterozygous type B and heterozygous type A. Then show the possible outcome of the cross. So we have here IAIB, heterozygous type A, heterozygous type B, two small i's. It is possible because the blood type of both parents are heterozygous for type B and type A. The table summarizes the various blood groups that children may inherit from their parents. Genotypes are shown in the second column and in small print for the offspring. Then the text above them summarizes the outcomes. Now this time, let's have polygenic traits. This inheritance pattern is sometimes called polygenic inheritance. Poly means many. A polygenic trait is a characteristic, sometimes we call them phenotypes, that are affected by many different genes. Polygenic traits require more than one gene or allele to determine trait. Polygenic traits are quite different from the classical Mendelian trait, in where we see that one gene controls one characteristic or one phenotype. Surprisingly, most traits in humans, and in fact, most traits in most organisms, are polygenic. Mendelian traits, although we spend a lot of time talking about them, are really the exception. Most genetic characteristics or traits are controlled by many genes. And here are some examples. You have A, hair color, B, skin color, C, human height, and D, eye color. Hair color. Same genetic code is found on chromosomes 3, 5, 10. The more dominant alleles, the darker the hair. Letter B, skin color. Skin color is now believed to be a polygenic trait governed by 3 sets of genes. Differences in skin color arise from the quantity and distribution of the pigment melanin in the uppermost layers of the skin. Exposure to sunlight is an environmental factor that can affect the skin color. Skin color is another trait that is very obvious in humans that is controlled by many different genes. And this is why you get differences between parents and children in skin color, although they tend to resemble one another. Next is human height. Height in humans is very strongly genetically controlled, but there are many different genes that control height, and this is why people are not the exact height of their parents. Variation in human height is caused by genotype and can be influenced by the environment. Example, a person has inherited a number of dominant allele for tallness and has the potential to grow tall. However, if the diet is poor, the body cells do not have sufficient nutrients for maximum development of the growth potential. And last is eye color, that is determined by 2 factors. Eye color happens because of the amount of melanin in the iris. A lot of melanin gives brown eyes and less melanin gives green. Little or no melanin in the stroma of the iris gives blue eyes. Varies from light brown to black depending on the amount of melanin in the front of the iris. And there are no genes for any color other than brown. Let's dig in. Blood groups exist as 4 different types, but are determined by 3 alleles, A, B, and O. Group O, Group A, Group B, and Group AB. Stage 1, the alleles A and B are codominant. The allele O is recessive to both A and B. What are the genotypes of each blood group? So for Group O, we have O, O. For Group A, we have A, O, and A, A. For Group B, we have B, O, and B, B. And for Group AB, we have A and B. Next, Try a cross between heterozygous parents with the codominant alleles. What genes are present in the gametes? We have here A, O, and B, O. Next, what are the possible genotypes of the first generation offspring from this cross? So we have here D, O, and A, O. They show all the possible outcomes. So A, B, B, O, A, O, and O, O. Stage 4. What are the possible phenotype of the first generation offspring from this cross? Group AB, Group A, Group B, and Group O. A cross between heterozygous parent with the codominant blood group alleles produce all 4 possible phenotypes due to codominance. Number 2, complete the Punnett square for a father who is blood group O and a mother who is blood group AB. The blood type of the father is type O and the blood type of the mother is type AB. So the gametes of the father is O, O, and the gametes of the mother is A, B. Then show the possible outcomes. We have here A, O, B, O, A, O, and B, O. What's the probability of having a child with blood group A? So the probability is 50%. What is the probability of having a child with blood group B? 50%. And what is the probability of having a child with group O? That is 0. Well done! Let's remember, in multiple allele, there are more than 2 alleles controlling a gene pair. More than 2 phenotypes are exhibited or expressed in the offspring. There are 6 different genotypes and 4 different phenotypes for blood groups. In AB blood group, the A trait and the B trait appear together in the phenotype. There are 2 genotypes for both A and B blood groups. In either homozygous condition, IAIA or IBIB, or in heterozygous condition, IAI or IBI, with one recessive allele I for O blood group. Blood group O appears only in homozygous condition, and polygenic traits produced by more than one pair of genes result in a variety of phenotypes. Let's apply. To gain more understanding about multiple alleles, these activities are provided for you. Read and analyze them well so that you will arrive on the right answers. Again, there are 2 ways in writing the genotype of blood types. You may use either of the given genotype in our discussion. Number 1, if a male is homozygous for blood type B and a female is heterozygous for blood type A, what are the possible blood type of the offspring? Number 2, is it possible for a child with type O blood to be born to a mother who is type AB? Why or why not? And number 3, a child is type AB. His biological mother is also type AB. What are the possible phenotype of his biological father? You may pause the video to answer this. Let's evaluate. Now let us try to evaluate how much you have learned about multiple alleles. Try to identify which among the sentences are facts about the pattern and which are not. Instruction: write T if the statement is correct. If false, change the underlined word to make the sentence correct. You may pause the video to answer this. So that's the end of our lesson. I hope you learned something new today. Watch out for the next video lesson in science. Don't forget to like, share, and subscribe. This has been your teacher, Rosalie Custodio, saying, learning science is fun, cool, and awesome. Thank you for watching. God bless.